So welcome everyone to our NextGen Precision Health Discovery Series. It's a great pleasure to introduce Dr. Anke-Hilse Maitland-van der Zee. So Dr. van der Zee is a expert in precision medicine of respiratory diseases and uses omics approaches and the interaction between omics and phenotype to understand how to improve health in patients affected by respiratory disorders. So she's a full professor at Amsterdam University Medical Center and is a lead principal investigator, or maybe the lead principal investigator of the Precision Medicine for More Oxygen initiative. This is a really cool initiative that is building these collaborative relationships between industry and academics to accelerate improved respiratory care and health. Very aligned with what we hope to do with NextGen Precision Health, the initiative, and here at the building. And so it's a great pleasure to hand off the mic for the title and talk, talk entitled Precision Medicine and Respiratory Disease, from Biomarkers to Building Consortiums. Welcome.
Okay, thank you very much for the very kind introduction, and thank you for having me here. I'm very much impressed by a whole building on precision medicine. We don't have that. Really cool to see, and I'm also really curious to learn what you guys are doing here. But first of all, of course, I will tell a bit about what we are doing. These are my conflicts of interest. So as was already told, I work in a public-private consortium trying to move prevention of chronic respiratory diseases forward, and therefore we work with big pharma but also with smaller companies. So there's lots of companies there that are part of our consortium and are also contributing in cash or in kind to our consortium. And furthermore, I also got some other grants from industry, pharmaceutical industry, and I did some consulting, and all the money was paid to my institution. I do not think that any of this is going to influence what I say today, but I just want to make sure that you're aware of that. So that's the title slide of my talk. So I always think it's nice to start off with telling why this is important. Why would we do precision medicine? And that's actually because of these two people there. It's because there's patients that still have lots of complaints and problems, even though they're treated according to the way that we've defined as the best way for treatment. So there's this little girl having asthma, and she's getting a beta agonist and an inhalation corticosteroid, and according to the guidelines, that should help her. But still, she's sitting inside watching her fish instead of playing outside with her friends, which is not what we want. So we want to figure out what's going on with the individual patient, and if the general therapy doesn't work, what can we do to actually help these patients? Well, the same holds true for the guy with COPD that you see on the picture. So I think that's why we're doing this. So I don't really have to explain precision medicine here, probably, because all of you work in it. But I think it's important to say that we like to think about the P4. That's also the field P, is why our consortium is called P4. And because I think it's very important that this is not just about personalized, but it's also about predictive, about trying to figure out who's at risk and trying to prevent things from happening, right? If we can do that on a personalized basis, very important. And then the last thing is participatory. And I know Professor Bartlett for a long time because we teach a course together at the International Society of Pharmacopoeia. I don't even want to think how many years, quite some years. And together with us, we are teaching with Bruce Carlton from University of British Columbia. And he always tells this story about a pediatric patient with cancer that's going to be treated with a drug that might influence hearing as a side effect. And then one day there's this patient that's blind. And what happens to you if you're blind and you lose your hearing? Then you lose all your contact with the rest of the world, right? So this adverse effect all of a sudden becomes far more important to this patient. So when are you going to what risk are you going to take to this patient? Are you going to give this medication, or might you want to choose for something else, particularly for this patient, because this patient shouldn't get this? So I think not only taking into account the risk of the medication itself, but also the rest of the patient, right? And involve the patient in that decision. What kind of risk do you want to take? So precision medicine, in my opinion, is far bigger, right, than just looking at genes and say, well, you cannot take this because you are a slow metabolizer or whatever. It's far bigger than that. So that's what I just wanted to emphasize. So this is a paper that I just read, and I just had to include it in this presentation because I really liked it, and I think it's telling us where to go in the future. And the author, Subaya, argues that we are not making use of all the techniques we have now enough, right? He says, we've seen during COVID-19 that we are so able to speed up the process of getting a vaccine to the market, right? If we compare it with all the other vaccines that have been developed, this was like a crazy quick procedure. Why was that possible? And why is it not possible for all the other things that we do? And so part of that can be realized by precision medicine, right? We are having, let's see whether the pointer works. Oh, well, it doesn't really matter. But we have all this extra information. We have omics data, we have electronic health records, we have AI techniques that we can use to mine through all this data. We have new designs for clinical trials, right? We are using the same clinical trial designs for 30 years now, but it takes too long, right? So we need the new ones, the ones that are taking into account that during the process you might want to change, that take biomarkers into account, the basket trials, the umbrella trials, all these kind of things. So making use of our new methods is going to help us speed up the process of drug development. And also, this picture comes from the same paper, we are only looking at the tip of the iceberg, right? Right now, we're only looking at the information that's clearly available, the trials that we use to come up with our standardized ways of treatment. But there's so much more data that there is, right? Like I said, the omics data we have of the individual patients, but we also have now the Internet of Medical Things, right? So we have watches that patients wear that can tell us about their heartbeat, that can tell us about their oxygen levels in their blood. We have there's so many data that we can bring into this process, and then we'll be able to do evidence-based deep medicine instead of evidence-based medicine. And I think this is what precision medicine is all about, and this is what's going to change the future and is going to get us out of the delay where we are now, where everything takes so long. So yeah, that's why I wanted to start my presentation with this. But I also want to show you some of the work that we're doing in my group. It's going to be a lot about asthma, because we do a lot of work on asthma. It's going to be about volatile organic compounds that you can measure in exhaled breath. It's a very nice, non-invasive way to find biomarkers for detecting different phenotypes of disease. And it's going to be about microbiome and transcriptome, because that's a lot of the recent work that we've done, and how this can help us to move towards precision medicine. And then Jill also asked me to talk a bit about our P402 consortium, on how we set it up, how we work with that. So that's what I'll do at the end of the talk. So starting off again with the asthma. Why is it so important to do precision medicine in asthma? Because actually what we call asthma, we don't think it's one disease, right? There's people with similar complaints, but having different biological backgrounds behind the disease. And if we're able to figure that out, we'll also be able to better treat them. So here is a very simple graph, just putting eosinophilic inflammation against symptoms. And if you think about it, the most logical form of asthma is the diagonal, right? The more inflammation you have, the more complaints. But as you can see, there's also patients that have a lot of inflammation, but not so much complaints, or even the other way around, not so much inflammation, but still a lot of complaints. For example, the obese non-eosinophilic phenotype of asthma. So only looking at these two factors, we can already realize that there's different phenotypes that might need different treatment, right, to be helped. So I think this is a very clear picture of why we need to figure out what's going on behind the clinical symptoms. So I already said I wanted to tell you something about the breathomics work in my group. This is just some of the people that contributed a lot to this work. Anirban, Paul, Marije, Mahmoud and Rianne. You'll see a lot of their work coming by, so I like to mention them in my talks. So first of all, why would we want to measure volatile organic compounds in exhaled breath? Well, because we're interested in the lung, and this is the air that directly comes from the lung, right? So what do you expect in this air? You expect, for example, metabolites from bacteria that live in your lungs. You expect metabolites from inflammatory processes ongoing in the lung. But also we'll measure things that come out of circulation, because of course there's a lot of contact between the blood and the lung, right? So there's also compounds coming out of the circulation. So it's not just what you have in your lungs that you measure. And of course there's also the air that we breathe in our lungs, right? So as we sit here, we'll have a lot of similar compounds because, I don't know, there might be still some salad dressing in the air or something like that. So it's very important to realize that it's not just all coming from the inside, but also, of course, from the outside, what we're measuring. So we've got different ways to measure what's in this exhaled breath. And the two main differences are that it can be pattern-based. So we use sensors that can measure a combination of different compounds that have a certain threshold, right? And then you get a pattern, and you can compare, for example, patients with a disease and without a disease and see whether the patterns are different in these two. And we can also use gas chromatography, mass spectrometry, to really look at all the different compounds that are in there, right? And then hopefully by knowing the compounds, it will be possible to also make a distinction between different things. We've moved a lot from pattern-based towards chemical-based because we figured out that the pattern-based was sometimes really hard to sell because a lot of companies or a lot of people that want to use this in clinical practice want to know what they're measuring. And if you can only measure a pattern, it's very much a black box, right? And really hard also to validate. So what we think we should do, what we are working towards now, is try to figure out the compounds that are important and then go back to the sensor. So find the sensor that can really measure these compounds because the sensor measurements are quick and easy, right? So for example, we found an important compound for Pseudomonas infection. We do also a lot of work on CF, on cystic fibrosis, especially in children. You want to know whether they get a Pseudomonas infection, you want to treat that. And so if we have that sensor in a very simple thing and it just becomes red or green, right? If it's there or not there, it can be a very quick measurement, non-invasive, especially in children, super handy, right? They don't like all these invasive procedures. So I think the combination of the two, that's I think where the future is, to be honest. But just showing you some results from our work in exhaled breath. So here we made a large database of all patients with different chronic respiratory diseases, and here you can see a combination of the patients that had asthma and COPD. So for who's not so familiar with respiratory disease, so most of the time asthma is a disease that starts in childhood, is very much inflammatory and also allergy based a lot of the times, while COPD usually starts at an older age, patients that have smoked or have been exposed to, I don't know, certain professional things like, for example, cleaners or bakers. Or, so there's a big difference in age in the populations, and in asthma, the problems with the lungs are usually reversible. If you give those patients beta agonists, they will be able to get more lung function, but that's not happening in the COPD patients where it's not reversible anymore. Anyhow, it seems like two very different diagnoses, but if we look at this, we combined the asthma and COPD patients in our cohort, and then we clustered them on how much alike they were on these VOCs, right? So the clusters you see in the picture, there was a training set and a validation set, and so we got similar clusters, but those clusters were, when we compared them, defined by the amount of eosinophilic, neutrophilic inflammation, the amount of A2P, the BMI of the patients, the packed years smoked, ethnicity, and exacerbation rate. But none of the clusters was only asthma or only COPD patients. And I think that's one of the things that shows that we are putting labels on disease, right? Very clear, you have smoked, so you have COPD. But of course, this person that has smoked might also have an allergic constitution, right? They might have an asthma component, and so the label might be wrong. So I think that's why it's so important to understand what's biologically going on. So in that same database, actually coming from the same paper, just to show you what we can do with exhaled breath, we also looked at the amount of blood, neutrophilic and eosinophilic inflammation. And here you can see that we could actually make dose response curves of those different forms of inflammation, just making use of exhaled breath. Without going too much into the details, because there's still a lot that I see. I'm talking too much again, like I usually do. So here you can see the results from a study in 4 different populations where we looked at patients with and without high IgE, so with or without A2P, to put it that way. And you can see that we could also smell that, right? That it was possible to smell the difference between those patients that had A2P and patients that did not have A2P. And we were also able to replicate that in different studies. And then the last results slide on the exhaled breath. here we had a population of patients, some were healthy and some had asthma, we actually exposed them to a virus challenge. So we gave them a virus, and then we looked what changed in their exhaled breath. And as you can see, so the upper row are the healthy people, the lower ones are the asthma patients, and the blue are the pre-challenge values, and the orange or pink, whatever you want to call it, are the after-challenge values. And then the different columns are different sensors that are in the electronic nose that we used. And you can see for most of the sensors there's a difference pre and post, and the difference is actually much larger in our asthma patients. And we thought, if this is true, that might be extremely convenient in clinical practice, because that means that we might be able to smell the fact that they were exposed to this virus and they might be getting ill before they feel it. And if that's the case, then we might be able to earlier treat, right? And maybe prevent exacerbations. And exacerbations are bad, because if you get them, sometimes your lungs even deteriorate more, right? So you really want to intervene quickly if possible. So that's why we also thought, and Paul Brinkman, the engineer in my group, is working on this, we need a very simple e-nose, like I already said before, that just has the sensors in there that smell what we need to smell, that they can actually have at home. And this is the prototype. I can tell you it already looks different because he's working on it all the time. But the idea is, and that's still the case, that we can put in different sensors. So the different little things you see in there are sensors. And so for the kids with Pseudomonas, I will need a different sensor than the adults with asthma, where I want to predict an exacerbation. So by making it this way, I can easily adapt the system for the patient that it's used for. And we hope in the coming months to bring the first 25 prototypes into the patients' homes to actually test. So this is really cool, I think. And it's also one of those things that's in the Internet of Medical Things, right? Because it can be connected with the computer, you can blow in it once a day or two or three times a week, whatever, and you can gather all this data and also compare with yourself, right? Because that's one of the important things. If something changes, that's when we need to do something, right? So if it's stable, it's all fine, and then something changes, and that might be the moment to interfere. So that was the exhaled breath story. I can talk much more about it, but I think this was enough for today. But if there's any questions later, I'm very happy to answer. So the other stuff I would like to see is on the microbiome, and this is Mahmoud Abdelaziz. He's a postdoc from Egypt. He did his PhD in my group. He was my only PhD student who got his PhD cum laude. He's really the smartest guy I ever had in my team. Mahmoud has got 2 PhD students, but I couldn't find pictures of them on the internet. So you saw some results, but I still have to ask them for their pictures. So anyways, talking about the microbiome, again, I don't know how much you're aware of therapy and asthma, but actually last years have been very good for severe asthma patients, because we got many biologics onto the market. Mepolizumab, omalizumab, tezepelumab, reslizumab. So there's a whole bunch of new medication available for these patients, and it works very well in a large part of them. That really changed the disease. But these things are mainly working for those patients having a lot of eosinophilic inflammation. But we also have patients in the asthma field that have a lot of neutrophilic inflammation, or not so much eosinophilic and other things. And for them, so you can see that in this picture, there's not so much treatment available. So we need to figure out what's really going on in these patients, right? So to also give them a therapy that they respond to, and make sure that they can live their lives as they want to. So if we analyze our data, and I'm going to show you microbiome and transcriptome data here, there's different ways that we can do that, right? We can compare our controlled patients with our uncontrolled patients, for example, and see how they differ. So that's more the classical approach, right? Where we compare and then look. But we can also just look at our data. We can look at the microbiome and say, okay, which patients are very much alike for the microbiome, which have the same bacteria, and let's make clusters, right? And then look at the clusters and see how they compare for the clinical characteristics. So we did both things in the coming slides. So the first studies I'm going to show you are out of the UBioPred study. UBioPred was a project that started now, I think, 15 years ago or something. It was an Innovative Medicine Initiative project. This is a European project, also a collaboration between industry and academia. And they included a lot of asthma patients different severities, and both adults and children, and they sampled all these different things. So they did a lot of sampling, and they did all the omics that you see next to it. And at that point in time, that was really innovative, right? That was really new to do that. And the data is still there. We published a really nice paper last week in the Blue Journal. I'll show you some results from it, from this data. So even though the consortium has no funding for, I think 8 or 9 years now, it's run out of funding, we still collaborate, we still have monthly meetings with the people in the consortium, we still do analysis, and we still use the data to write new grants. So having really good data and very well phenotyped people, extremely important. This is one of the papers we wrote. So we looked at the severe adult asthma patients in the Biomarkers consortium. There's 2 measurements in them, one at baseline and one 12 to 18 months later. We looked at both time points. And we did, like I said, the unsupervised microbiome clustering. And so when we did that, we discovered 2 clusters. So this is the visual abstract from the Yaki. And in the one cluster was much bigger than the other, and the smaller cluster was the more severe cluster. And in the smaller cluster, there was more airway obstruction, there were more pathogenic bacteria in the microbiome, there were more neutrophils and a bit less macrophages. So hey, here we have them again, the neutrophils, right? So this is partly the population where we don't have a lot of treatment possible. So in the first cluster, there are more of the commensal bacteria, more the ones that you would expect. And then we also looked again at them, this 12 to 18 months later, and a lot of them stayed in the same cluster. So it seemed to be very stable where they were. So just a bit more data on this. So these are the clinical characteristics. And so you can see the difference in the neutrophils, right? It was significant. You can see that there was a lower lung function in the more severe cluster, as we expected. Also a lower lung function after salbutamol, so also the reversibility was a bit less. And this is, like I said, 85% of the patients stayed in the same cluster, so at the second time point. So that means that it's not just something at one moment, but if you measure it a long time later, you see it's still the same way. So it's important to know. So within these 2 clusters, we then looked at the transcriptome. So the transcriptome of these same patients, and we looked whether there were differences, and there were, right? And many of those things that were different are actually in the neutrophilic asthma signature. So again, we come back to these different phenotypes that we actually already knew, and now we found them again by looking at the microbiome. And here you can also see the pathways that were up and down regulated. And without going into all the details of that, it can help us, right? If we understand which pathways are involved in these patients, we can also think about treatment. We can also think, how can we intervene in these pathways and help these patients? So this is going to help us to identify subtypes. It's going to help us to find therapeutic options. We now know that something in the microbiome might be the problem, so maybe treating with azithromycin, for example, an antibiotic, can be helpful for these patients that have the problem. We can look for other therapeutic targets. Now we start to understand the pathways in these cluster 2 patients. And also the neutrophilic pathway, again, is not just one thing, right? Again, there's different mechanisms, different pathways involved. And if we know them on an individual basis, this can help us to treat precision medicine, to treat the individual patient the right way. And one of the things, this was a trial where they were looking whether etanercept was working, and their conclusion was that it didn't work. But we think they looked in the wrong population. So again, right, smarter clinical trials try to figure out which biomarkers you need to measure to figure out for which patient, which treatment will actually work. So there it can also help. So that was the adult work. I also like to show you some of our work in the pediatric cohort. So this was the paper that we published last week in the blue. Again, so here we're looking at a pediatric population with a mean age of 6 years, but quite a large age range actually, from wheezing kids until kids in their teens. And most of them were Caucasian, and then they were either having severe asthma, mild to moderate asthma, severe wheezing, or mild to moderate wheezing. And this is mainly based on age, right? Because under a certain age, it's really hard to measure lung function. It's also really hard to get a real asthma diagnosis. So it means the young children are in the wheezing group and the older children are in the asthma group. And we also know about their antibiotic intake, and around 12% had current antibiotic intake. Very important if you study the microbiome, because of course that's really changing the moment that you give those kids antibiotics. So try to summarize it for you here. We found 4 clusters within the pediatric, and they were different on the amount of eczema, the amount of atopy they had, especially grass pollen. They were different for lung function, post-bronchodilator, and for the risk of exacerbations. And so our second cluster actually had the most atopic, had the most eczema, and also had the highest risk on getting exacerbations in the coming 1.5 years, which was followed during the Biomarkers study. And again, we looked also at pathways, and we also found that there were 2 different pathways important, and that the Wnt, the signaling pathway, was very important in our 4th cluster, while in the 2nd cluster it was far more the TGF-beta pathway that was important. And both of them have been found important in remodeling in asthma, so in changes in the lung that are not very favorable if you get them, to put it that way. So different mechanisms behind it again, and again we identified those by looking at the microbiome. And this is microbiome in throat swabs. So you can do the microbiome in a lot of different things, but this was throat swab. So quite easy to obtain from children, because again, you need to think about invasiveness, right? Can you do it to them? But this is not something that was extremely invasive. So here, we didn't talk much about the bacteria yet, but here you can see the bacteria, the different colors are the different clusters, and the main bacteria on 2 different levels. But if we look at the bacteria itself, you can see that cluster 4 mainly has Haemophilus as the main bacteria, while Rotia is the bacteria that's main in the cluster 3, Veillonella most in cluster 2, and Streptococcus in cluster 1. These were, and if we were looking at those different bacteria, and we took the upper one, we were classifying a really large part of our children in the right cluster. So this also means that maybe we don't have to do a full RNA-seq, right, on all the bacteria, but we can just look at a couple to determine in which cluster they belong. And also the Veillonella, that was the group that had most complaints, right? And this was already shown in other studies before. That's a really deleterious bacteria to have. Well, the Rotia bacteria has been shown to be protective, also in other cohorts. So it's also makes sense, right, the bacteria that we found in these different clusters and the clinical characteristics that went with them. So here you can see the pathways that I said before, so the Wnt pathway and the TGF-beta pathway, and that they were differently expressed in those different clusters, but both of them were playing a role in remodeling in asthma. And this is actually, I think, one of the nicest things out of this paper, is that it actually can help you to predict, right? So looking at in which cluster a patient is will tell you something about their risk for developing exacerbations. If you are in cluster 1, you have hardly any risk on exacerbations, right? Well, if you are in cluster 2, you have a pretty high risk on getting more than 2 exacerbations in the next year, which is a pretty big thing for kids. So it really means that we need to do something for those, right? We need to figure out how they are treated. We need to see whether we need to do something about this bacteria that are present. It's really a thing to continue our work on. I mean, we published this last week, but I think this can have next steps. This is going to help us determine who to treat and how to treat in the future. So that was in Biomarkers data. Then we also set up a systems pharmacology in pediatric asthma consortium a bit later. That's now around 6 or 7 years ago. It took us a long time, especially because of Corona, to get the children in. But we have 145 children in. They were included in 4 different countries in Europe. And there again, we sampled quite a lot of things, and we did a lot of omics. Here you can see that it was in Spain, Slovenia, Germany, and the Netherlands. And here I wanted to show you some gut microbiome data. Until now, we've looked at the upper airways, right, the throat, but here this is gut microbiome. it's interesting because we don't always directly think about this, but the microbiome in our gut is extremely important for our health because these bacteria make something called small chain fatty acids, and they go into our system and they do influence our immune system, for example. So it's very important to have a healthy gut microbiome. And we also compared children in this Sysfampedia study. And again, we did it two ways. We looked at the controlled and the uncontrolled ones, but we also just looked at the bacteria and tried to come up with clusters. So this is the classical approach. It was a case control study, so the kids were included because they either had complaints or exacerbations in the last year, or they did not, and they were all in the same treatment category, so they had a similar severity of asthma that way. And if we looked at the volcano plots, you can see that we didn't find anything significant if we looked at differences between our cases and controls. And then we thought, well, this is a very basic way of looking at it, but why don't we use a bit more fancy, a bit more bioinformatic approach? So let's go for machine learning algorithms, and we did with the help of some bioinformaticians. And then I think the upper plot is the saliva and the lower one is the feces. We had two kinds of microbiome samples in those kids. And for both of them, we could distinguish the cases and controls when we were using machine learning. So also the method that you use has an effect on what you can find. And here, again, so the microbiome does determine which kids were controlled and uncontrolled. And then in that same data, and this is for the gut microbiome, we also did the cluster analysis. And here you can see the two clusters that we found. And here's the data on the clusters. Cluster 1 was bigger than cluster 2. The BMI Z-score was higher in cluster 1 compared with cluster 2. There was also a difference in the country of inclusion. You can see that most of the cluster 2 patients came out of Spain, for example, and there were no cluster 2 patients in Germany. Also interesting. Then there was a difference in the amount of eczema, amount of food allergy. The allergic history of the mom was different so it says something about the genetics. Then the history of antibiotic intake was different. The frequency of severe asthma attacks was different. More in cluster 2, again the more serious one, more ICU admissions in cluster 2, more eosinophils and more basophils in cluster 2. So yeah, I think again a more severe and a less severe cluster. And here you can see the differences in the bacteria in these two clusters. And here it was really remarkable that in cluster 1 there's mainly Prevotella. So that's the one that you see all the way on the top, the most abundant in cluster 1. And then in cluster 2, we had the most abundant were the Bacteroides. So again, what Mahmoud did was also look at whether he could predict in which cluster someone should be just by looking at those two. And it was a very high, again, a very high way of predicting by looking at just those two bacteria. So I think once we've done all this analysis, we don't always need to do the complete omics analysis right later on. We can also have some markers that tell us what this probably looks like. And the other thing that we thought was interesting was that already earlier publications, like this one in Science, talked about two kinds of patterns, enterotypes in the gut, and those two enterotypes, this comes right from this article, were actually either Bacteroides or Prevotella dominated. So these were actually the two same bacteria that we found in our asthmatic population, there was so this is interesting to see that we find the same ones, and this has influence on your severity of asthma, and again might help us in finding optimal ways of treatment. So we can use non-invasive biomarkers to cluster patients to find optimal treatment. We can identify those patients at risk for exacerbations, for more severe disease. And we can also think about new leads, and especially in the T2, in the patients with the neutrophilic asthma, that's really important, right, that we find new leads. But we shouldn't forget it's not just about medication, right? It's also about lifestyle advice. It's also about healthy life. BMI is very much related also to microbiome, gut microbiome. Living a healthier lifestyle can also help you adjust these things. So let's always keep that in mind. And then also the e-health thing. I think this is the future, right? We measure things at home. If we can measure these volatile organic compounds at home and we have a direct link with the physician when something changes, this can also optimize treatment by just being earlier, right, in intervening. So that was my talk about our recent research, but I also promised to tell a little bit about P402. So I have 10 minutes for that. I can talk about P402 for hours, so I won't, no worries. But we set up P402 a couple of years ago. It came actually out of an initiative from the Netherlands Respiratory Society, where the two Dutch gurus of lung research said, you know, we're doing so many interesting things in lung research in the Netherlands, but we should collaborate more with each other. So if we bring all the researchers, but also all the physicians and patients and companies together, and we sit in a hotel in a distant place where no one wants to be, so everyone stays inside, then maybe we can identify what we should do in lung research in the Netherlands in the coming 10 years. And there were 3 topics identified, and one of those was precision medicine and prevention. And so that's how we started to set up P402. I was one of the people that started that initiative, and they said to us, you know what, make it big. Great, how do you make something big, right? We're used to writing grants, right, and you get it or you get it not, but here the idea was go and talk with the companies, go figure out what people want and try to build something together. Well, that was quite a long process. It's almost 10 years ago that we had this first meeting, but we managed. We wrote a proposal, we got in many companies that wanted to do this with us, and there's a Dutch organization called Health Holland that's there to motivate public-private consortia. So they gave us a large sum of money, and together with the contributions from our private partners and from our universities, we had a budget of 14.5 million euros for 4 years, and we'll have this until the end of next year. So what we decided to do was build cohorts, healthy cohorts, high-risk cohorts for chronic respiratory disease, and cohorts of patients with this disease. So we and measure the same things. So measure the same biomarkers, measure the same omics, measure the exposome, what are these patients or healthy people exposed to, and try to find markers to earlier diagnose, earlier find out who is prone to develop disease, and hopefully also find ways to intervene in that. We also work with in vitro models. We've got several groups that make lung on a chip, nose on a chip, muscle on a chip, and we connect these models and we try just to, the danger of talking too much, but what is so nice within the consortium is that we have these very basic researchers, we have the epidemiologists and the clinicians and everything in between, and we measure a lot of exposome. We go to the patient's homes, we give them sensors, or no, actually filters, that collect all the dust, all the PM2.5 they ran into. We weigh it, and then we know exactly, you know, how much they ran into. We do that for 3 days, 2 times in the year, because in the seasons it might be different. But then also what's in these filters we freeze. So we have the actual exposure, and then we use that frozen stuff in our cell work. So we can expose the cells exactly to that, you know, contamination, to that problem that our people are exposed to. And I think that, right, being able to make that link between the very clinical and the very basic work, that's what makes me very enthusiastic. Anyways, these are the academic medical centers in the Netherlands that are involved. Honestly, there's only one missing, and we're talking with them now. We're getting them in, because that's a bit pathetic, right, if they're not in. But apart from that, all the others are in. These are our private partners, so ranging from Big Pharma, Boehringer Ingelheim, Roche, AbbVie is in it now, they just joined, to very small companies that make diagnostic devices. Speak, Breathomics, they make different e-noses. Also companies that do AI on CT scans. We make CT scans of all the patients, and we think we can learn more from it than just looking with a physician's eye, right? Making use of AI. So a very broad range of companies is in our. That's also why my conflict of interest is so long, right? So this is what I told you actually already. So these are the work packages. We now have a long COVID cohort, a healthy high-risk cohort we're building, the COPD cohort is ongoing. We now have funding for an ILD cohort, interstitial lung disease. We are submitting it to MRB this week. We submitted some grants for a pediatric cohort, which wheezing kids are going into asthma. And we're talking with Lung Cancer the Netherlands, who really wants us to include a lung cancer cohort as well. We do exposome in all the patients, imaging, CT scans in all the patients, and omics. We have an AI work package, of course, to deal with all the data that we get. And then there's the cell culture models and trying to get as much interaction as possible between these different things. So this I think I already said. We do nasal brushings, breath, blood, urine, and faces. That's what we collect samples. These are the omics layers that we do. So I already talked a bit about the exposome. Very important, very new to me, right? We did so much omics. I started with pharmacogenetics many years ago. We went into genetics, we went into all the other omics. But only part of who we are and how healthy we are is by our build, right? It's also a lot of what's coming from the outside into us, the exposome. So looking at that, I think is very important. So we try to link all our data to the national map of the Netherlands, right? There's data on traffic, there's data on pollen, you can link, if you know the postal code, it's possible to link. So that's one thing that we do. We also take biological specimens, so we can measure metabolites in urine, for example, for things that they've encountered. And then we have these activity trackers. So I already told you about the UPAS sensor, that's the thing that they wear for 3 days. We also have sniffer bikes. The idea was to put them on the bikes of these people, because you might not know, but Dutch people like to bike. The only thing was we started with our long COVID cohort, and those people were so tired, they were not biking. So then we decided not to do that, and we put them stationary out of a window outside their house. And we leave them there between the two measurements with 6 months in between, so we have 6 months data of PM2.5. And I've seen the first data, and for example, there was one time there was a desert dust storm in the Netherlands, so if the wind is really bad and comes from Africa, right, we got all of a sudden, and you can just see that peak, right? And the other peak was with New Year's Eve, when Dutch people tend to do their own fireworks. That's what they like. So I mean, I like to see that because it's like really clear that if there's things ongoing, you will see it in that data. So we have that, then we have the silicone wristbands where the toxic compounds can be absorbed, and we have Garmin trackers just to get their exercise, their physical activity. So because we're working with private companies, one of our companies is called LogicCare, and they are a database company. And so they make the database for Coca-Cola and for KLM, right? For hospitals, but now also for us. So we get this every week. So this is just one of our long COVID cohort, but it tells us how many study visits have been done, the first one, the second one, how many exposome visits have been done, what's the age distribution. Well, you can put in here whatever you like. But I think this is really nice, also showing off, if you have these companies, you have things that you wouldn't have if you were just doing it as an academic institution. I'm going to be exactly within time. So this project is ongoing. First results are out, first papers are out. And it's so nice to work with this big group of people. We are so enthusiastic. We've just submitted a grant for 77 million euros to the Dutch government because we're trying to make this sustainable. If we get it, we'll start a company and a non-profit organization. The things that we are discovering now, we've got a long list of possible products. We will try to bring to the market using the company. And if the company earns money, it will go back to the non-profit organization and it will flow back into the P402 research. So if we can make that work, it should become sustainable in the end. That's the idea. That's my group in my garden. We also have green in the Netherlands. It's really nice and green here. I've been driven around this morning and I really enjoyed this here. That's the CIS1-PDA Consortium. And of course the main investigators in UBioPred, but that's much bigger than that. But then the slide would be too full. So. And this is the picture of the P402 Consortium meeting. So I wanted to end with that and very happy to answer any questions you have.
Did you have any twins in your studies?
Twins?
Twins, yes. And if you learn anything from asthma, microbiome, and so on.
I don't have twins in my studies. We do collaborate sometimes with the Dutch Twin Register. They only have twins, so we have done some work in a twin register, but in our studies they're not there. But you're right, it's an interesting population to look at. I agree.
Or siblings. Have you studied like a control?
No, we don't do that, no. We don't have that in our studies. But if you want to control for genetics, of course, that's an idea. And that's why we do like to work with the Twin Register. But if you look at their asthma data, then of course they not always have everything we need. But it's definitely an interesting way to look at things. We haven't done it for microbiome yet. It's mainly genetics what we've done in the Twin Register.
Thank you. Thank you for the nice talk. I have a question. You talk about several clustering analyze, and I was wondering if you have the same technique for all of them, because you know that the clustering, the performance of the clustering depends on the data and type of the data that you have. And if what kind of a clustering did you use, the machine learning technique or the mixture model?
We have used a lot of different techniques. I'm not a bioinformatician, I'm just a simple clinical pharmacologist, but I do have several in my team, which is good. And they, so I can, for all the different, so they use different clustering techniques for all of the different clusters that I showed you. And then by looking at that, they decided which was, the best number of clusters and the best one to use. And I can give you all the details on all the different ones, but I don't can do that.
I just wanted to see if yes.
Yeah. No.
And my other question is that in one slide you talk about the baseline cluster and the longitudinal clusters, the transition metrics. Did you look at the transition metrics for different treatments and see that if the cluster transition is different for the different treatment?
No, I don't know if you've seen the numbers, but they were quite low, right? It's like 85 patients or something. Yeah. So I think that would probably be very hard, and it's only 1 year, right? So it's not a very long period. So we were just curious to see whether if we looked at the same patients again 1 year later, whether they would be in the same clusters or not, but we didn't link it to anything else. I think actually for us the main thing was that whatever happened to them, however they were treated, and they were in the study, so maybe they got more attention, but still 85% was in the same cluster, and we thought that was the main thing there.
Thank you, very interesting. Thank you.
So I know a bit more about what you've done to put together these consortiums, but for everybody here, can you talk specifically the P402 consortium? How often do you need to meet together, and what's been sort of the biggest hurdle for you coming from academia to make this happen?
Well, I can start with answering the easy part, which is how often we meet. So you've seen the structure in the work packages that I showed you. So we have meetings for the different cohorts monthly. So the COPD, the healthy, high risk, long COVID, and now also for the ILD, that's every month. And then and also for the in vitro work has their own monthly meeting. And then next to that, we have what we call the project integration team meeting. So that's people from all of these meetings coming together to make sure that we all know what's going on in the other meetings. That's also monthly. And then we have a bi-yearly project committee meeting, and that's actually what this picture is from. So twice a year, we all get together in person. The others are online. Because of the travel time and only 1 hour, but this is a one-day meeting that we have twice a year. We think it's extremely important to meet. It's extremely important to talk, and the only way to get everyone's input is to bring people together. And of course, not everyone's there every time. That's simply impossible. But these are the places where it happens, right? Hey, I've got these dust samples and you've got these cells. It would be great, right? My exhaled breath people are now measuring the air that goes through the lung on a chip model. So together with the long-on-chip people, they managed to get a method, even though it's very small amounts of air that go through there, to sample that air and to look at the exhaled metabolites of cells, which is really nice because then you don't have all the disturbing factors, right, that we have around us. So, but this happens because we bring those people in the same room, the companies in the same room. They also start to collaborate. So I think that's one of the important things. And hurdles, yes, there's always hurdles, right? If you tell it backwards, it seems like one big success story. It wasn't. I mean, we had companies promising us 600,000 euros, and then after even signing letter of intent, all of a sudden calling higher management decided we can't do it. You know, things like that. These are the days that you're not so happy. You know, we don't know whether we get the Dutch grant. If we get it, you know, we'll be very, but we're already thinking about, you know, what are we going to do if we don't get it? These people like to collaborate. And they've also seen great things coming out of this. And then if that momentum is there, you need to continue and you need to think of all kinds of ways to keep it going. And also do fun things. Or young investigators, the PhDs and postdocs, I think there's 18 of them in there. They visit the companies. They get lectures from the companies. They visit each other's labs. The guys from the in vitro lab went to the exposome lab and they were very surprised about what they saw. So that as well, right? Let them do fun things together, have dinner in the evening, talk. Communication is key.
You mentioned 4 or 5 times Pseudomonas aeruginosa as a bacterium involved in this. And the testing that you use to detect these bacteria with mass spectroscopy and gas chromatography. Have you explored anything like UV fluorescence that is typical for pyoverdine and pyocyanin, which are kind of iron chelating extracellular products of this?
I did not, to be honest. I mentioned Pseudomonas just a couple of times because it's, and I didn't show it here because I only showed asthma work. This is cystic fibrosis, right? In asthma, thank goodness, we don't see a lot of Pseudomonas issues. In CF patients, we do. And it's a very clear issue, right? This one bacteria causes a lot of problems and you want to know whether the patient is infected or not. So it's a very clear one, and we published a couple of VOCs that are important in, you know, signaling this particular bacteria. But there's probably other ways to do it, and UV light might definitely be one of them. I'm not very much into that, to be honest. But especially for children, this is a very easy thing, right? You blow in something and then it gives you an answer. So that's why we were focusing on that, but there's probably definitely many more ways you could do it.
At least in urine, if there's some signal of infection, the urine is lighted with UV, and you can see, notice that infection without having the bacteria.
Thanks. I didn't know that, but good to know.
Thank you so much for your talk. Really fantastic. I have 3 questions. One's pretty easy, one's middle, and one's maybe a little harder. And you don't.
We still have 8 minutes,
and
so
you
I
don't have
tried
to
to
answer.
talk 8 minutes about the first one.
Exactly. You don't have to answer all 3. But the first one, I was curious. I really love the interaction between exposome and disease. In the in vitro approaches, are there any of the folks using like patient cell lines, so iPSCs, where they convert? So then they're also looking at genetics. Is that part of the approach, or are these just standard cell lines?
I think they're doing both, but again, I'm an epidemiologist, not an in vitro person, so I'm not the right person to answer your question, but we are both using cells from our own patients. So for example, in our long COVID work, in my group, we have immunologists that do the no-cell work, and they use the cells that come directly from the patients. Also in Groningen, they make the long on the chip. I know they use cell lines, but we do also want to use the cells of the individual patients, because that in the end is the whole idea of P402, right? To optimize treatment for an individual patient. So I think we do a lot of the development work with the cell lines, and then once we're trying to see whether something works, the actual idea is to have the cells from the patients and try it first in vitro before we try it in the patient. So use it as a way as an organoid, right? As a system to try and try out therapy.
Yeah, I love that crosstalk between the different approaches to answer a question. I mean, that's beautiful, and hopefully we can do that here. So the next question, you briefly mentioned antibiotics in the gut microbiome. How about in the upper respiratory? And what are your thoughts on diet and antibiotics? How does that influence the microbiome?
It does.
Yeah.
It does
Yeah.
a lot. So what we try to do in our cohorts as well is ask patients to write down what they've eaten for a couple of days. I mean, I'm a pharmacoepidemiologist by training. You know, analyzing what drugs people take is complicated, but it's nothing compared to food, right? Because, well, we all don't eat exactly the same food every day. What we report might not be what we actually consumed because, well, you know, sometimes we are a bit sorry about what we did. So it's really hard to measure, and it's also a lot of work, right? You don't only want to people to write down what they ate, but also how much. And so we've seen that the data is not always very good. We're starting the healthy cohort very soon, and there we're collaborating with another university in the Netherlands that made an app asking for what you ate the last 2 hours. And it will ask for that many times. So instead of asking them to write 3 full days, we'll go for a longer period and every now and then send them an app and then ask them, okay, what did you now? So you don't have to remember as much, you don't have to write as much. They say that it works better, so we'll try that. So having an adequate information on the food is not easy, but very important. We've tried in Cis-Pharmpedia and the Pediatric study that I showed. We've got also the food diaries from the kids, and we have small chain fatty acids, both in feces and in blood, and we have metabolome. And we're trying to link those 3 layers, but linking the food with the 2 others is really hard. So again, it's probably because we don't have the right, I think we just don't have the right information. So it's really hard, but very interesting. Food and drugs, of course, as well. Yeah, we also, of course, try to get all the drugs that they use. But,
One more question. So very excited to hear about your future plans. So you're talking about defining a lot of targets and approaches, and you're refining those, and I love that. So as you develop this company, or maybe you already have mechanisms in place, like intellectual property, IP, patents, and like the business model with this industry academic partner, like how do you navigate that?
it's really interesting, very good question, and a very difficult question.
That was a difficult question.
I know, I understand that. So we thought a lot about this in the past months because we submitted that grant. It was February 3rd, I won't forget because. But anyway, so there we collaborated with, I actually found a partner in crime who set up several companies himself. He's a physician by training, but a very commercial guy. And I needed him because I'm an academic, right? And this is just really new stuff to me. But so what we, so in the Netherlands, a lot of this is playing in the Netherlands. So if there's an invention at an academic institution, the institution is the owner of it. And so the institution can have the negotiations with the company. So the company that we will start, it's not there yet, will negotiate with the universities, and we will, our idea is that it's probably not going to be these separate interventions that are going to make the difference. Like for these multifactorial diseases, we think you need more diagnostics to figure out all the phenotypes, and you need different interventions to solve the problem. So what we want to do is bring together a couple of diagnostics and interventions and say, okay, if we do all this, and we call that a target product profile, then it can do what we need it to do. And we also are negotiating with the healthcare insurances that if we make something that can do what we've defined, that then they will pay for it. So they already tell us that they will pay even though they haven't seen the product. Because that means we can get investors. Because the investor likes the idea that later on it will be paid for. So coming back to the university, if we can say, well, it's not just your product, but in combination with those 3 other things that we get from other universities, so we will pay you this, but the rest, is the company bringing those things together. So that's where we think the margins for the company are, bringing this stuff together. We don't want to produce things. It's not going to be a company that makes a drug or a diagnostic. The company is going to bring it further, help it develop within academia, and then sell it to pharma or help it become a company itself. right? A spin-off company. And we already have investors that are very interested to support these spin-offs.
Great.
So the company will earn the money and bring it back. So there's been a lot of thought about this.
Thank you
I don't
so
want to
much.
bother everyone else with it, but if you need more, then we can
Yeah,
talk
we should
about it.
maybe follow up and talk about that. It's a little bit more complicated in the US, I think, with how IP tracks with the institution, the PI, and so forth. But thank you so much.
I make it quite simple. I know there's more, but I think in the end, if you cannot bring it to the market by yourself, then at least you can earn something, by doing it in combination. And I think, at least Dutch universities are willing to talk about this.
Great. Thank you so much for your talk. Really appreciate it. And so I mentioned last time, we do have a unique format for the next Discovery Series. So we're going to have a live viewing here by video, and then we're also going to be at UMSL. So it's going to be Dr. Winograd. So please join us.